You will give a conceptual insight into understanding of histology of spleen, the largest secondary lymphoid organ which also is filtering the blood. Splenic histology is all about the parenchyma. Then the parenchyma consists of white pulp and red pulp, which we will understand first by a schematic diagram. So here we have a capsule of spleen, and beneath the capsule we have a space that is trabecula. This trabecula is what contains trabecular artery. Spleen is supplied by a splenic artery which branches off to a trabecular artery which is present in this trabecular space. Trabecular artery gives off a central artery which is the main artery supplying the parenchyma of the spleen. This central artery is surrounded by a group of cells. These group of cells surrounding central artery are T cell. Now central artery branches off to a central arteriole. This central arteriole is again surrounded by a group of cells, but this time these are B cells. Splenic arteriole eventually gives off capillaries, and this part of the circulation is closed circulation since the blood is flowing within the vessels. Now these capillaries, rather than draining into veins, are draining into spaces, and these spaces are known as sinusoids. These spaces are actually surrounded by something. These are cords which are surrounding these sinusoids, and these cords are what are known as cords of Billroth, which are made up of cells and red connective tissue. Cords of Billroth is also known as splenic cords. So this part of circulation where blood spills off into these sinusoids consists a part of open circulation. Now via these splenic cords, the blood in the sinusoids is drained via tributaries into vein, then into trabecular vein, and trabecular vein eventually drains into the splenic vein. This is how circulation of spleen is working. Now this part of closed circulation which actually had central artery surrounded by T cell is periarteriolar lymphoid sheath, and another group of B cell surrounding a central arteriole is what is a splenic follicle or lymphatic follicle. So white pulp is a part that consists of the closed circulation, and white pulp actually has periarteriolar lymphoid system and splenic follicle, and these both form a part of immunity via cell mediated immunity as well as humoral immunity via B cell. Now the part of the splenic circulation which had sinusoids and cords of Billroth is what is red pulp. Sinusoids and cords of Billroth. These part along with macrophages help in the filtering of the blood. Let's see the histology image now. So we have here number of follicles. These follicles are what are the white pulp. These follicle can be either the splenic follicle or periarteriolar lymphoid system based on the cross section we have taken. Now we see these follicle have a special thing that we see here. These are the arteriole or the artery, that is central artery or arteriole. Now the location can be centric or eccentric based on the proliferating cell. If it is actively dividing, we will see a eccentric location of the central artery or the arteriole. If it is less dividing or less active follicle, we will see a central arteriole in the central. This is the part which will eventually drain via the capillaries into an open circulation. That is the main part of parenchyma we see. This is the red pulp. Red pulp, though not so visible red, but we see that here we have an open circulation and we have the splenic cords as well as sinusoids that we saw in the image. Let's see another image here. So here we see white pulp. White pulp is a lot of basophilic because of lot of nucleus of T cell and B cell. And here we see the area where we see the spaces sinusoids which are filled with this blood, RBCs. And here this is the area where blood is being filtered in the spleen. Another white pulp area, another red pulp area. So splenic histology is all about understanding the circulation of spleen. Also see this image. What we see is a central artery surrounded by a group of lymphoid cell that are T cell, and this is what is known as periarteriolar lymphoid system. Notice how PALS and splenic follicle are different based on the artery or arteriole they are surrounding and the group of cell B or T cell they are surrounding. So this is all about histology of spleen and what you needed to understand. Thanks for watching.